This vodcast is about the she statements for the 2023 Year 12 ATAR biology course. Now, the first thing we'll talk about is artificial selection. This is where humans decide upon the desirable traits for a species. It's not left to nature to find the desirable traits. And what we mean by desirable traits are the mutations that are created naturally, and they try to retain these and enhance these in the population. They choose which individuals, those individuals that have the trait, they allow them to breed. Those without the trait, they are prevented from breeding. This process takes many generations to occur. The effect of this is an increased expression of the trait in the population. When we say an increase of expression of the trait, we mean there are more individuals in the population that have the desired phenotype. As we said before, it takes several generations to occur, therefore it's considered slow. Changes the genes for the entire gene pool, reducing genetic diversity because we are selecting which alleles are allowed to be inherited in future generations. And eventually it's hoped that the entire population has the traits desired by the humans. Now, the traits desired by the humans can be purely for looks, such as the French bulldog, or it could be something more advantageous to us, such as a cow that makes more meat, a cow that produces more milk. It could be crops that have an increased yield. It could be fruit trees that have bigger fruit and more fruit, or it could be something as innocuous as an albino guinea pig that wouldn't survive in the wild, but we like to breed our albino guinea pigs because they look nice and the children love to pat them. Okay, next is transgenesis. Transgenesis just means taking the genes from one species and putting it into the genes or the genetic code of another species. First of all, you need to identify the gene that codes for the particular polypeptide or trait that you desire, cut it out with restriction enzymes, perhaps amplified with PCR or gene cloning, and then put it into the recipient's DNA. And then when you mix two types of DNA from two different species, this is how you create a genetically modified organism. So a GMO just means that you've had DNA from two different sources put into the one place and therefore into the one species. Now, it doesn't need to be related. Proof of concept was achieved by taking the luminescence gene out of a jellyfish and put into things like mice and fish. This can occur because DNA is the universal genetic code of all life on Earth. So you can take the DNA from any species and put it into any other species because they all have DNA as their genetic code. And we say that the DNA code is universal across all species. Right, transgenesis is a little bit more difficult to achieve than something, say, like artificial selection because you need specific biotechnology tools like PCR and gel electrophoresis and restriction enzymes. Transgenesis gives you results a lot faster than artificial selection because it's immediate. You take the genes out of one species and put it into another species and you get an immediate effect. Because you are deciding upon the alleles/genes that you want to have in a different species, you are in effect reducing the genetic diversity because all the individuals after that time are going to have exactly the same DNA, and this greatly reduces the genetic diversity of the species. Examples of where this has occurred, we have golden rice. We have found the gene or the allele that makes vitamin A, and we incorporate that into the rice plant, and so when the rice plant makes rice, it also has a lot of vitamin A, which brings vitamin A into countries that have a very poor diet and a lack of vitamin A. Another one is Bt cotton. We have the bacteria Bacillus thuringiensis that makes a natural pesticide that when an insect eats the leaf, it ingests a pesticide that kills the bug that's eating the plant, thereby increasing the profit from the crop because there hasn't been as many bugs eating the crop. Now, the ways to get genes into other species, well, we know about the TI plasmid. TI plasmid comes from the bacterium Agrobacterium tumefaciens. The TI plasmid stands for tumor-inducing because the agrobacterium snuggles up to a plant cell and then it sends out a tube that goes through the cell wall into the plant cell, and then it's got its TI plasmid that it transfers and then it inserts its genes into the genome of the plant cell, and we just utilize the technology that's already been invented by the plant. Another one is the gene gun, where we put pieces of DNA, very small pieces of DNA, say alleles or genes, onto a speck of gold, and then we fire that speck of gold into the nucleus of the host cell, and the DNA then gets incorporated into the host genome. And then finally we have vectors. Some of the most efficacious vectors are things like viruses that are naturally able to inject their DNA into a host cell by using enzymes that are also injected, are able to put their DNA into the host DNA. And one of those really important enzymes is reverse transcriptase that turns RNA back into DNA that then inserts it into the host cell's DNA. DNA profiling allows us to compare populations and individuals within a population. DNA profiling involves the non-coding regions of chromosomes that used to be called junk DNA. We use STR, which stands for short tandem repeats, which are short repeating units of nucleotides such as GATA. When there is one repeat, it's just GATA. When there's two repeats, it's GATA, GATA. When there's three repeats, it's GATA, 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 and so on. As we get more, or as we increase the STRs, or the repeating short tandem units of DNA, the fragments will get longer. We know that there are about 10 to 15 loci for STRs in the human genome, and the more loci we use, the more accurate the DNA profile becomes. So first of all, we find a restriction enzyme that we know cuts out the STR fragment from the chromosome. And a good restriction enzyme to remember is EcoR1 that comes from E. coli that cuts at the palindromic sequence GAATTC. So we use a restriction enzyme to cut out the STR. We then amplify the STR fragment to increase the numbers of it. We know that we can stain it with a lovely stain called Midori green. We then run it through a gel, and then the banding pattern gives us what we call the DNA profile. And then we can compare the DNA profiles of individuals within the same population and between different populations to look for relatedness. For recombinant DNA technology, we need to identify and isolate the gene of interest. We cut it out with a restriction enzyme, such as EcoR1. We get a plasmid out of a bacterium, and we cut it with the same restriction enzyme so that we get the same sticky ends. We then get the isolated gene and the cut plasmid and tie them together with DNA ligase. We then put the recombined plasmid into a bacterium in a process called transformation. We can do this by thermal shock, which is to raise and lower the temperature, or we can shock it with electricity, and that will cause the bacterium to take up the recombined plasmid. The recombined plasmid is then put into a lovely growth medium at a lovely pH and a nice temperature, and it undergoes binary fission to increase the numbers of the bacterium that contain the recombined plasmid, and then the bacterium makes desired protein. Gene cloning is very similar to recombinant DNA technology. We identify and isolate the gene we want to clone. We extract it with restriction enzymes. We cut open a plasmid using the same restriction enzyme and tie it together with DNA ligase. We then insert it into a vacant bacterium in a process called transformation. We then allow it to undergo binary fission, and as it undergoes binary fission, it copies its DNA, and that increases the copies of the gene required. We then harvest and isolate the DNA, and this results in many more copies of the desired gene that we wanted to copy. Now we can use all of these biotechnology tools to protect biodiversity. What we need to be wary of is small populations or inbred populations that have reduced genetic diversity. If there is reduced genetic diversity, these populations can't cope with environmental change. Natural selection only sees the phenotype of an individual, not the genotype, and with reduced genetic diversity, there is not a lot of phenotypic diversity. And when the population gets smaller, none of the individuals will have a selective advantage, and the population will be prone to extinction. This is where biotechnology comes in. We can use DNA profiling to monitor the gene pools. Remember, DNA profiling uses restriction enzymes, PCR, and gel electrophoresis. We can tell if the population has a low genetic diversity by comparing the DNA profiles of the individuals within the population. We can also stop inbreeding by not allowing closely related individuals from breeding. means we will select individuals for captive breeding that are unrelated. We can put them into a zoo environment and allow them to breed, and their offspring will not be inbred. We can also bring in individuals that are genetically diverse from other populations to increase the biodiversity within the population. Recombinant DNA and transgenics can be used to protect biodiversity. We can bring in genes from other species that have an advantage or increase the genetic fitness of the species. So we would find the gene that provides the selective advantage. We would put it into the individuals within the population that's struggling with change in the environment. Examples of this are allowing crops to become drought tolerant, allowing crops or trees to become salt tolerant. So this increases biodiversity because crops or trees that would normally die under these changes are allowed to live, thus increasing biodiversity. We can use artificial selection by selecting the genetically fit individuals from a population that are known to have a selective advantage, and by artificially selecting or choosing which individuals can breed, we allow the population to survive and even cope with the environmental change. We even have frozen zoos and seed banks, which are pretty much the same thing, except a frozen zoo has frozen embryos and a seed bank has seeds from various plant species. The advantage here is meiosis is used to make the gametes, and we all know that meiosis increases genetic diversity through crossing over in prophase 1 and independent assortment in anaphase 1 and anaphase 2. Once we've got the embryos or the seeds and we grow them up, we can replenish wild populations with reducing genetic diversity, and then we just need to make sure that we put them into an area with reduced disease and or reduced predators, and this will allow the population to flourish.